So welcome to tonight's webinar. My name is Bill Thornburg. I'm the AMTA Executive Officer. So it's good to see you. Thanks for giving up your Tuesday night to spend some time with us. I want to read a little intro about your presenters for this evening. You have Dr. Ingrid Waldron, who's a professor emerita at UPenn. Ingrid has cooperated with colleagues to develop biology lessons and provide PD for high school and middle school teachers in the Philadelphia area for years. Her biology lessons, including the detailed teacher notes and student handouts, are and they're available on the Serendip website.
Come here.
You have Larry Dukerich there, a longtime modeler. Larry taught high school chemistry and physics for over 30 years. And as one of the early modelers, he's been working on developing curricular materials in physics, physical science, and chemistry, and has conducted numerous workshops in physics and chemistry since 1995. And finally, Ray Hoansky, who's the Director of Curricular Development for Ridley School District, which is in the Philadelphia area. After teaching high school chemistry and biology for 22 years, he moved to the administrative side, and he's helped in the development of several of the instruction units in the modeling chemistry curricular materials. And in addition, has facilitated many modeling chemistry workshops. I was one of his students back in 2010 in Indianapolis. Ray and teachers at Ridley High School enlisted the help of Dr. Waldron and Larry Dukerich to modify Dr. Waldron's lessons to develop a modeling biology course that would serve students in schools that use the physics, chemistry, biology sequence. So this course builds on the chemistry background that students have from their previous course, and it's well aligned with the NGSS disciplinary core ideas and science practices, such as using multiple types of models to provide mechanistic accounts or predict phenomena and move flexibly between model types. So this webinar tonight is intended to provide an overview of the materials that have been developed in the direction of future work. So you are in great hands tonight. First thing, if you have that chat window, you'll see that Ingrid, Ray, and Larry put their emails. If you missed those in previous communications, there you have them. If you've just joined and you missed me say it earlier, I continue to add resources to the AMTA COVID-19 emergency resource button, which you'll find on our website, modelinginstruction.org. With all these biology minds, if you're using anything that you have found to be fantastic for you or your students about certain resources, how to virtually teach better, labs and activity demos, whatever it is, if they're great and you want me to share them with the rest of the modeling community, please send that to me at my email address, which I put in there, and I'll be happy to get that posted on the site. So the chat window is going to be open. I'll be monitoring it. If it's something that's really, really important, I'm going to get their attention and they're going to address it. But they also have planned question time, question and answer time at the end. So I'm going to turn it over to Ray, Larry, Ingrid. You guys have fun, and we'll see you in a bit.
Okay, so this is Larry. I want to make sure that you understood. I'm hoping that you are able to see a screen that's got a PowerPoint presentation and a Word document. If that's true, sort of raise your hand or whatever so that I know that I'm not going to leave anybody behind.
The only thing is if you could make the PowerPoint just without the frame.
I can do that. You mean like that?
Yep.
All right. So I do want to make sure that you know that this PowerPoint, the overview document, the individual topic overviews will be available in a Google Drive folder that we'll share with you, and the address will be on the final slide. So you don't need to, if we go too fast or something like that, you can always come back and look at it. And Bill is recording the session. So what we wanted to say, and you did hopefully get this copy of this overview document in an email since you had registered for the meeting. What we're going to do is walk you through a little bit about what we're trying to do in this treatment. So I'm going to scooch down to table 1. We want you to understand that the course really tries to identify and articulate those in a course designed for a physics, chemistry, biology sequence. So the models are here, and there's a description of each one of those as we go along. And it's a very thorough treatment. I think you'll recognize that maybe after a bit of a beginning that's heavy on the chemistry, just to help set the stage, the topics that are addressed are pretty much what you'd find, I think, in most any solid biology course. Now, we don't have all of these done yet, but we have everything done through the first page. So,
It like shut off. It didn't have any.
I'm sorry. I didn't hear that. So I'm just going to, so this is a summary of the overview. It explains what the course is intended for, and explains how we make use of the students' understanding of chemistry as they go on to learn about DNA, transcription and translation, basic molecular biology, structure and function of cells, genetics, and evolution. So I am going to show, let's see, here's where we are right now. We have 5 topics that we have addressed. 3 of those topics have been broken down into 2 instructional units each, like biological molecules has the chemistry of life and then enzymes. And we end up with a treatment of energy in biological processes. Unit 1 talks about characteristics of life and levels of organization in biology. And I'm one who doesn't like to read PowerPoint slides, so I'll just pause long enough for you to take a look at what you see there. It's the intro unit that I think that would be common to many courses in biology courses that are taught around the world. Now, topic 2 moves into biological molecules, where we build on what they've learned. Remember, this course is a capstone biology course, so it follows chemistry, where students have learned about the properties of these essential elements. We talk about the properties of functional groups, small molecules, how these molecules are combined to make more complex macromolecules, such as proteins, and we understand how these biological molecules can help us understand cells and organisms. I'm going to show you next objectives for, first of all, Unit 2A, give you sort of some sense of what it is we attempt to accomplish in that unit. So we talk a little, about these properties. We want to be able to understand why is it that some molecules are polar and nonpolar, how molecules interact with one another. We talk about functional groups in organic molecules, identifying amino acids, fats, nucleic acids, and carbohydrates. we talk about how these smaller molecules, which are monomers, combine in repeating patterns to form polymers. And then we talk about what some of these things are: proteins, fats, carbohydrates, and nucleic acids. Skip that for just a second and go on to define objectives for, oops, where are they? Here are the Unit 2B objectives. Here once we've talked about assembling amino acids into proteins, many of these proteins function as enzymes. We talk about the role of enzymes and how their structure affects their properties and behavior, not just at the molecular level, but it actually has implications for organisms. So we talk about specific cases where people suffer from enzyme deficiencies, such as lactose intolerance. We discuss the role of energy in these reactions, and this is where we have an opportunity to introduce energy bar charts. Something I neglected to mention in the last, in Unit 2A, we end that particular unit with an experiment that provides students the opportunity to generate hypotheses from observations and to test and subsequently revise them. So it's the introduction to scientific methodology. I'm going to pause right now and give you guys an opportunity to ask some questions.
There's one in the chat box, Larry.
All right. And I have to see if I can get back to that because I'm showing my, when I'm sharing my screen, I can't see that.
Larry, the question's from John, and he said, how is Unit 2A in the bio related to Unit 12 in the chemistry storyline? Is there overlap or repeating of information? Or does 2A build on Unit 12?
I'd say, so I'm going to stop sharing my screen for just a second here, and then I'll pop back into it. Unit 2A mirrors Unit 12 in the chemistry curriculum. So it would certainly serve as a review of what they learned there. It builds on it in terms of talking about some biology-specific instances. Does that help?
Yeah, that's great. Thank you, Larry.
You bet. Anything else before I move on to turning this over to Ray for his portion of the talk?
I just want to mention that not everybody is muted, and we need everybody except the speaker to mute themselves.
Thank you for mentioning that, Ingrid. It may be that people who joined a little bit later didn't get muted by Bill. Okay, I'm going to go back to sharing the screen. And Ray, it is now your turn.
Thanks, Larry. So I figured I would wait to add on to Larry's answer there about the Unit 12. You know, we very much had that in mind as we developed the start of the you know, biology capstone course here. So you see a lot of similarities and connections, and since the same folks were involved with developing it, I think it'll be easily recognizable. So as Unit 2, topic 2 emphasizes the structure and function of the molecules and how they support living systems. And specifically, DNA and proteins are part of that treatment. We kind of pick up in Unit 3 with one of those specific molecules discussed, and that would be DNA. So what's different about the DNA treatment here is in large part where it is placed. As it points out here in the slide, we strongly recommend that this come after the treatment of the chemistry of life and prior to the unit that Ingrid will be following up with, that is cell structure and function, that topic. So,
You want to see worksheet 1?
yes, please. So we will kind of build upon the understanding of both Unit 2 with the structure and function of those molecules, and specifically DNA and its structure and function and how that affects what's occurring at the cell level in genetics and evolution. And we do this in such a way that students can refer back to their understanding of these molecular interactions to explain those other levels of organization of living systems. So the basic organizational principle here is that we talk about or students see and experience DNA as the code of life through the DNA molecule and its structure, and how its structure facilitates its function, which is to produce proteins that have a specific structure, which establishes certain functionality in those proteins, which ultimately leads to the characteristics that living organisms have. In Ingrid's materials that we build upon is that link of the DNA to the protein to the characteristics, with no black holes left for students to wonder about. I think that is a strength here. There is mechanistic explanations from one level to the next, from the molecular level to the organism level, and I think that is something that sets these materials apart. Now, prior to this worksheet, just to kind of go back real quick, the unit does start with an experience, which I think is.
I think we need to get everybody to mute themselves.
I think somebody's trying.
And Larry, can you mute people if they don't mute themselves?
I believe I can.
I just did. It's whoever the Fireflies is. There's a lot of noise coming from there, so I muted them.
Okay, I'm sorry for interrupting, Ray, but
That's okay.
I'll keep an eye on that.
All right, so okay, so now train of thought.
We're ready for worksheet 2, Ray.
So we're getting ready to worksheet, but before we go into worksheet 2, let me just real quick go back to the opening of the unit. We skipped over that the students do extract DNA in a lab experience, and one of the really nice of that lab is the fact that because of some of their background in chemistry, there's an explanation provided that's accessible to students so that they can understand each of the rationale for each of the steps in the extraction of DNA. And they really get a strong sense of the fact that DNA has to be accessed through membranes, and that we need some molecular interactions in order to free that DNA out of the nucleus. So I think that's a strong part of the, that opening lab experience. Then on to worksheet 1, which gives you that general connection between the DNA and its functionality producing proteins, which ultimately then leads to those characteristics in the organisms. And then in worksheet 2, Larry, if you can pull that up. So in worksheet 2, and go down just a bit, please. So there's that with the sequence, shows the sequence, the chart with the sequences. It's down more. So right there, so that's fine. So, so while in worksheet 1 helps students frame their understanding of the connection between DNA and proteins and the characteristics of life, this worksheet really helps the students understand that the sequence of the nucleotides in the DNA is important, right? And the function of that is to determine that sequence of amino acids in proteins. And the function of that is to determine the structure and function of those proteins, which then ultimately influences those characteristics of the traits in the organisms. So kind of going from a broader framework to one more specific, and again, building on the molecular understanding of how the interactions of those molecules, mechanism of one level of organization influence the next level of organization, in this case, from the molecular to the cell, and then ultimately to the characteristics of the organisms. And if you can kind of focus in on question 5 there, this really gives an opportunity for students to think about, how this actually works. And that, the example of melanin, and that's the dark pigment of skin, and then they actually look at the differences of the nucleotides and the genes that could result in the characteristics that you see in the picture there. So just if I may just pull back for a second and highlight that the examples that Ingrid has selected that we work with are also simplified as possible, but yet rooted in real examples. So that too, just like any good model, it's as simple as possible to demonstrate or to explain the phenomenon that we're talking about. And so the selection of those examples, as well as the selection of diagrams, as well as the selection of the language that we use, is really important to distill down, the complexities of reality into something that's digestible for high school students. Then, in worksheet 3, we go through DNA replication, in which the students see that, okay, we understand this process by which, proteins are produced. But then it's also really important as reproduction occurs that those new organisms that are produced can also produce those same proteins, so that that organism can perform those same functionalities. And so that's really the emphasis of worksheet 3. And then you're looking at worksheet 4 on the screen. And just let me kind of highlight here that modifications of structure. So models help us to understand some aspect of complex reality. Way too complicated for us, to have, conversations with high school students, or let's face it, even for us, or any of us, that's what science does, right? We try to come up with better and better models, to approximate reality. And the better, those models are, and I think we're experiencing that a bit now. The better those models are, the better we can predict and work with work with the phenomena, whatever we're dealing with. In this case, we're dealing with the code of life, with the molecular code of life, with DNA. So we work with students and help them have experiences so that they can examine, reflect upon, and explicitly have conversations about decisions that are made in these diagrams, what they emphasize, what they de-emphasize, what they exclude about the features of DNA, and that these aren't just randomly produced, but rather very intentionally. Why are dotted lines used in one place and solid lines used in another? Why do some molecules, why are they designated as maybe something that you want to focus on and identify? So all these types of features are very consciously, decisions have been made very consciously to bring out certain features and characteristics. All right, so Lara, if you can go down to the second page.
And by the way, I want to point out we're already behind.
Okay. So this is a set of questions. I'll go as fast as I can. So this is just a set of questions. And if we're looking at the replication model here, that the geometric shapes, right? So they're made very intentionally, and that the molecular structure of the phosphates and the sugars are illustrated specific ways to illustrate and highlight certain features of the DNA molecule that allows the function in its replication. Okay, so
I'm going to switch over to the Unit 3B teacher notes.
I think we're ready for 3B and teacher notes.
So we thought that one of the things we would indicate or show you, this is an example. The structure of the teacher notes is very similar to that found in the physics and the chemistry materials. So there are very robust learning goals, and you'll see that each of these units have explicit references to the next generation science standards, the DCIs, and the science and engineering practices. So if you have any questions about making sure that your course is aligned to those, all of that information is there and it's done for you. So there's an overview document, or a section I should say, and there are robust instructional notes that talk about what is it that emphasize on the reading, some background information for you, the teacher, to help you have a more meaningful conversation with your students. The activities have supplies, activity notes, how to conduct the post-lab discussion if necessary. I'm going to show you, there is one little episode in here where they do a modeling activity with ribosomes to show transcription and translation. And I am going to have to pop over to my so at the Serendip website, where there's a link provided here, I'm going to have to cover up a little bit of the PowerPoint. This gives you an idea where the original source of the materials is. I'm going to click on this little animation to show you what the activity would sort of look like in a sort of stop action, step by step.
And you'll notice that this shows the basics of the processes, taking out the complexities of the molecular biology and any unnecessary cell parts, really just focusing on the nucleus and the ribosomes. Models are only useful if they simplify enough for students to understand.
Yeah, so now how do I how do I quit out of this? There we go. I'm going to stop. And I'm going to go back to this. And I think at this point, this was the second portion of the slide that Ray was talking about. I don't think you need to see my Zoom thing here. Drag my browser window back to, there we go. And Ingrid, then do you want to start or do you want to give people a chance to ask some questions?
I'd rather start since we're running behind already.
That sounds fine.
So the cell unit, the first part of the cell unit is an introduction to cell structure and function. And students develop a model of the cell in which they learn about the size, the components, the differences between prokaryotic and eukaryotic cells. And one thing that's often not emphasized, we know about the diversity of eukaryotic organisms because we're familiar with that, but the incredible diversity of the chemical capabilities of prokaryotes is discussed in this unit with some striking examples. And then we do talk about the functions of eukaryotic membrane-bound organelles. There are three general principles that emerge from this part of the unit: that organelles work together to accomplish cellular functions, that structure is related to function. For example, that's illustrated by looking at red blood cells, sperm, and other types of human cells where the structure is quite varied. And another theme is that cells are dynamic with constant molecular activity, unlike the models, the diagrams that students really often see. So this is one of the worksheets in this part of the Unit 4. And you can see that in the upper figure, the students see how ribosomes, the rough endoplasmic reticulum, and the Golgi apparatus work together to produce proteins that are secreted. And then the next question, 5, has students develop a representation of the model of the cell by thinking of it as a factory and thinking how all the different cell organelles work together to produce proteins that can accomplish each of the functions shown. So now if we could move on to the next slide in the PowerPoint. We spend a fair amount of time on developing a model of the cell membrane because this is one of the clearest examples of how molecules interact to accomplish cellular function. And in the first lab, in this part of the Unit 4, students observe osmosis by looking at, putting eggs that do not have shells in a variety of solutions, some hypotonic, some hypertonic. And then they move on to worksheets where they analyze this phenomenon and understand it as an example of selective permeability of the cell membrane. And then they apply their understanding to animal cells, plant cells, and phenomena like water intoxication, where runners or athletes or people engaged in drinking water contests can take in so much water that they disrupt the neurons and can actually die from them. The next two experiments have students understand selective permeability of a synthetic membrane, where they make a hypothesis based on observing that many large molecules need to stay inside the cell and many small molecules like oxygen need to cross the cell membrane. And the second experiment follows a worksheet in which we remind students about polar molecules and their solubility in water, or ions insoluble in water, but nonpolar molecules not soluble in water. And there's a nice little experiment where they see that a layer of oil can be a barrier to diffusion ocean. And that's really a reasonable model of what the cell membrane, how the cell membrane functions.
Ingrid, I'm going to throw that picture up there for just a second.
Okay. The drop of food dye, which is an ion dissolved in water, just sits on the top of the oil for at least a little while, and then eventually it falls through. Then we have an activity where we introduce phospholipids, and you can see the structure of the phospholipid molecule here. The students are shown, are given a diagram which shows water molecules. Do you have that, Larry?
Yeah, I'm bringing them up. I got to find them.
Okay, so they're given the water molecules, drawing like this, and they know the water molecules are polar. They have the phospholipid molecules cut out from this sheet here, and they know about the charges on the phospho end and the nonpolar lipid tails, and then they're asked to figure out what, based on what they know about polar and nonpolar substances and attractions between positive and negative charges, how they would imagine these would arrange themselves, and then hopefully they come up with an answer like this. So that provides the basis for introducing the phospholipid bilayer between the watery exterior and the watery cytosol. And then we have worksheets where they analyze the roles of the phospholipid bilayer and membrane proteins in producing the selective permeability of the cell membrane. So that is all I wanted to say about the Unit 4. And if we could go on to topic 5. This is the last of the topics where we already have really well-organized teaching materials ready to use. In Unit 5A, students develop a model of cellular starting with an introduction to ATP as a key molecule in energy metabolism, and then using various representations of cellular respiration to develop a model of cellular respiration. And then students can apply this model of cellular respiration to, for example, how do muscles get the energy they need? What's the relationship between food intake and calorie intake and body weight and amount of calorie expenditure? The same progression occurs in the photosynthesis part, where students use various representations to develop a model of photosynthesis, and then they use their models of photosynthesis and cellular respiration to understand changes in light and in the dark with varying amounts of photosynthesis. So in the next slide, we move on to topics where we're going to develop the units. And the Serendip website that's been mentioned and that you've seen has the activities we'll use to develop this topic. So we begin with the analysis of mitosis. The students do a modeling activity to learn about mitosis and also use diagrams. Then meiosis produces genetically diverse haploid gametes. And then they use the model chromosomes to model meiosis and fertilization in a format that replicates, that duplicates what Punnett squares do, so that Punnett squares become a representation of the outcomes of meiosis and fertilization, rather than some arbitrary thing that geneticists made up. And we will return here to molecular biology and go into more depth. Now, topic 7, the students learn how the structure of organs and organ systems is related to their functions. And we do have an activity on negative feedback and how that helps us to maintain homeostasis. Topic 8, we hope to get through topic 7 by the end of the summer. Topic 8 probably won't get done before the fall. So the exponential and logistic population growth models, we explain, or we have the students understand how they derive from organism level processes. And we also give examples of where these models do and don't apply. And we go back through what Ray mentioned about models only work if the underlying assumptions of the models are correct. They're simplified and they apply to some circumstances, but where the circumstances are more complicated, see exponential or logistic population growth. When the students learn about food webs, again, we build them up, their understanding of food webs, together with photosynthesis and cellular respiration, to have students understand energy flow through ecosystems, carbon cycles, and trophic pyramids. For natural selection, we have a couple of different activities, including a model, cloth with pom-pom model of natural selection, and an analysis of the, what's the name of the moth? Biston betularia, whatever the common name is. And analyze several examples of evolution. Peppered moth, thank you. We analyze several examples of evolution. For example, how could something as complicated as the human eye have evolved? We use data from comparative biology, from DNA analysis, and from modeling, quantitative modeling, to help students understand how we can learn about the evolution of the eye, even though the fossil record is minimal. So I think we have time for questions now, am I right?
We do.
Okay, so let's look at the chat. So in response to integrating organ systems into the earlier units, we've thought about it, but it seems to work better to just postpone that, focus on the cells before that, and before that on the molecules. So I can see that Jim has been his efforts. Can you explain the hands up students model this? What are they doing in class besides the worksheet? So I'm not sure when that came up. We do have, as I've been discussing, experiments at various points throughout the various units. Do you want to say anything more, Ray?
I'm trying to figure out, like you, I'm trying to figure out where that question came in. That was right in the beginning. Maybe Larry's part there?
No, I don't think so.
I think it was maybe mine.
So Larry, I do see a question you can answer. Is there a link to all these materials?
Yes, that's how I plan to conclude. But I wanted to give people an opportunity to ask content-specific questions first.
So there's a question about doing bio in 9th grade. So there are other biomodeling activities available on the AMTA website, other modeling courses for biology. And the same modeling, but is used by many teachers who teach 9th grade biology.
Yeah, I think it's important to emphasize that this course was designed to provide a set of experiences that build on students' understanding of physics and chemistry. So it is intended to be a capstone course. But I will show, I guess I need to share the screen here again. I stopped because I was, so here's the final slide of the PowerPoint. And this gives you the URL to a Google Drive folder, which will have the PowerPoint presentation, the course overview that you've been looking at, overviews for topics 1 through 5, and when Bill gets done with it, he'll save it and then upload a Zoom recording of this webinar. And I guess I can take people to the modeling website to show them what it looks like when they go there, if we want to do that now.
I think it'd be good.
Yeah, and I do just want to point out in response to that one question about the hands-on activities that, and Larry and Ingrid, if you guys would respond to this as well, I think it's an integral part of each of the units is a laboratory experience and or some examples that engage students in modeling some phenomenon. So, you know, like obviously, you know, there's Ingrid has selected some examples that engage students in figuring that out, and then there would be some hands-on activities would challenge students to, if you will, model, you know, those phenomena. So they're integral in each of the units, you know, labs and hands-on experiences.
So if you log into the AMTA website, member website, which I just did, and I'm hoping that this is showing.
Yes.
Browser, and then you click on the instructional resources. Then you'll see you've got them for physics, chemistry, and biology. You click on the link for biology, and then you'll find the NGSS modeling biology capstone curriculum. If you select that, then what you'll see is the way it works at AMTA, each one of these things is called a record, or like a card in a Rolodex file. So there's an overview. Here are the first 5 topics that we were talking about. And if you were to pick, say, energy and biological processes, and you selected more details, then you get a summary and full description. And then here are the documents that you can download. Each one of the first 5 topics has an overview. 5A and 5B are zip folders. There's a unit test, and then there's a PowerPoint that we have provided that talks about the way we like to treat energy throughout all of the courses provided by AMTA. And I'm going to go back to, similarly, here's the topic 3. Summary and full description, overview, and then the zip folders for the units, which you can download. So I suppose what we can do now is open it up to questions. If you had a chance to copy down that URL, we'll send it to you via email. If you didn't, and actually what I can do, I should have thought about this. I will put it in the chat.
It's in the chat already.
John was quick on that.
You're good, buddy. You must be the executive officer.
No, John, I wish I could take credit. John did it.
So do you want to speak to the question about quantitative analysis of energy?
I didn't see that question, but
It just came up.
Okay. We deal with energy in a qualitative way. So the energy bar charts are qualitative, semi-quantitative ways of describing what's going on. Does that sort of answer the question? I mean, there are no experiments per se where we do calorimetry or whatever, because we build on that in the chemistry. Would this capstone biology course replace an AP biology? I am not competent to answer that question.
I think not. The AP course has, quite a specific set of things that students are supposed to learn. So I don't think so.
But I'll bet you it would be a very sound preparation for an AP course.
Absolutely.
Yeah. Ingrid, do you want to tackle this about the choice of sequence? Why we go from, the molecular level to the DNA, to DNA to then cells, from Colin Costello, 748. I can speak to a little bit.
I don't see the question, so go ahead.
In modeling chemistry, the storyline goes from observable systems and zooms into the atomic level, follows this discoveries. This biology sequence seems to start at the molecular level and zoom out. Curious to why the choice was made, and we did make that intentionally. And I didn't know if you and Larry wanted to speak to that.
I can speak to that. Since this comes after a chemistry course, and since DNA and the understanding at the molecular level is so central to every aspect of modern biology, it made sense to us to start by talking and giving the students a basic understanding of DNA, the central dogma, so that they can then apply that to cells and organs and organ systems and evolution and genetics. It's just a very central part of how biologists think about living organisms at this time.
Yeah, and it wasn't until like if we were looking at various levels of organization, you know, to not treat the molecular and the DNA, the central dogma up front, you really can't refer to that level of organization if the students haven't explicitly, you know, investigated that.
And most students will have been exposed to organism and ecology, biology in middle school. So they have a reference point to think about things.
So there's a question that said, thanking us for the presentation, that said, will you be presenting some of the other units beyond what we have done tonight? And I think the answer is to be determined.
As soon as we have them ready, I think we can do that.
Another question is, are the docs Google Doc compatible? I think that they're all in Microsoft Word, and so far I haven't had any difficulty uploading them into Google Docs. Now, of course, some graphics get lost along the way, but oh, somebody answered that question. John answered the question.
Yeah, and I would really encourage folks to dig into those teacher notes as well. Like, go into one of these units, and I think, you know, like we're painting with a rather broad brush here, but I think, you know, like Larry showed those teacher notes for Unit 3B. But if you really get in and take some time and read through the details of the teacher notes, I think you'll see that this is strongly supported, you know, with both rationale and background information that a teacher will find really useful.
Well, I'm going to have to say I was enlisted to help with the Unit 2, Topic 2, the chemistry of biological processes and the energy unit. But then I kept on with the other units, and I have learned an enormous amount of biology just reading the teacher notes. So I think that they really provide strong background for people of all different levels.
I wanted to say one more thing about the question of starting small. We do bring in human familiar examples already while we're talking about DNA and proteins and so on. So again, it links to a phenomenon that would be more familiar to them, to students. All right.
Well, it looks like we are 1 minute shy of running out of time. So do you have any closing words, Bill?
Nothing that's life-changing, no. Thank you all for, well, to the three presenters, thank you very much. And to everyone that showed up, thanks, and hope you got something worthwhile out of the talk. And then hopefully we'll get you guys to do another one and come back and visit us for an update on the materials.
And meanwhile, people can write us if they have specific questions.
And there's also a Google poll set up in the overview document that allows you to offer feedback.
Okay. All right. Well,
Thank you all
for coming.
All right.
Yeah.
Thanks.
Stay COVID free. Be safe out there.
All right.
Good luck with the rest of your semester. And Larry, I'll like you, I heard you say it. I will send this off to Wendy tomorrow, and we'll get it posted. And then if somebody, especially a non-member, if you have any non-members, reach out and ask questions. We can share it with them since this was open to all. So that's not
Okay, sounds good. You're still recording, I think.
I am still recording. And Tanya, are you there still?
Yeah, I'm sorry. I think you might have just answered my question. I am on the East Coast, and I thought this was from 8 to 9, but I must have been reading for a different time zone, so I missed it. But you just said that you did record this?
Yeah, it'll be posted on the ANTA website. Wendy's 6 hours ahead, so it'll be sometime tomorrow by the time she gets it up.
Thank you so much. I'm sorry about
No,
the.
that's okay. Sorry you missed it. But yeah, it'll be up there.
Okay.
And Tanya, feel free to send us any questions you have after you watch the recording.
I was looking forward to this for a long time, so I'm so bummed that I missed it. But I'll get your information and I'll take a look at what you got out there and figure it out. So anyway, thank you.
Okay, great.
Thank you.
Nick,
All right, I'll
If
go ahead
Nick's-
and I'll stop the recording, and you guys have a great night. Thank you so much.
You too.
And Ray, I'm going to send you an email or read Wendy's email so we can discuss the possibilities.
I will.